Hello, this is Stephanie Falck, a research technician for the USDA ARS, and I'm here to present this week's EOARC Ag Minutes. This week I'll be discussing the genetics of horse color. It's been said that a good horse is never a bad color, but everyone has their favorites. What is amazing, however, is that all colors, from bay to white to loud paint, start with two base colors, black and chestnut. All colors in horses start with this foundation. Black pigment in mammals is caused by eumelanin. In black horses, black is dominant. It can be present in either a homozygous or heterozygous form. Red pigment in mammals, called pheomelanin, is controlled by a recessive gene. This means that a horse must have two copies of the red gene in order to be chestnut. So this explains black and chestnut horses. But what about the most common color, bay? A bay horse starts off with the black pigment. The horse has an additional gene called agouti. This gene restricts the black pigment to the points of the horse, that is, the lower legs, mane, tail, and tips of the ears. Agouti is dominant. But what happens to a chestnut horse if they carry the agouti gene? Nothing, because although the agouti gene is dominant, a chestnut horse does not have any black pigment to be modified. So this explains the black, chestnut, and bay horses. This week I'm discussing the genetics of horse color. Today I will cover one of the diluting genes, cream. Dilution genes work by diluting the base colors we discussed yesterday, black, chestnut, or bay. The dilution genes are dominant, which means that in order for a foal to display the trait, at least one parent had to have the same trait as well. The most common dilution gene is cream. The cream gene is incompletely dominant, because it will only lighten the base color. If a chestnut horse has a single cream gene, it will be a palomino. A bay horse with a single cream gene will be a buckskin, and a black horse with a cream gene will be a smoky black, although the horse will still appear black. Genetic testing is necessary to confirm if the presence of the cream gene is there in the black horse's case. If a horse has two dilution genes, the colors are lightened even more. A chestnut horse with two copies of the cream gene will be a cremello. Cremellos always have pink skin and blue eyes, and range in a shade from light to very dark cream in color. In the past, they were thought to be albinos. However, they do have pigment in their skin. Scientists have not yet found a gene for albinoism in horses. Perlino is the color that occurs when you get two cream genes on top of the bay base color. The horse will appear cream in color with blue eyes and pink skin. However, the points of the horse will appear darker. Smoky cream is the color that occurs when there are two cream genes present with the black base coat. The horse will be cream in color. The only way to definitively know if the horse is a smoky cream is to have their DNA tested to see if they carry the black genes. Dun is another dilution gene. It affects both the black and the red pigment, but leaves the pigments of the points dark. Unlike the cream gene, it does not appear stronger if present in one dose or two. The dun gene causes primitive leg markings in horses. These marks are a dorsal stripe, zebra stripes on the legs, cobwebbing on the forehead, and shoulder stripes. Dun present with the chestnut color results in a chestnut dun. More commonly, you will hear these horses called yellow, red, or claybank duns. When the dun gene is present with a black gene, you will get a black dun, known as a grulla horse. When the dun gene is present on a bay horse, you will get a bay dun, which is usually commonly known as just dun. These horses are often confused with buckskins, because the colors are similar. However, if your buckskin has primitive markings, he's a dun, not a buckskin. Dun can also happen in addition to cream genes discussed yesterday. So a palomino with a dun gene is called a dunalino, and a buckskin with a dun gene is called a dunskin. Other common dilution genes are the champagne gene, which gives the horse a silvery color on top of their base color. Silver is another dilution gene that affects only the black pigment. It's responsible for the chocolate-covered horses that are often seen in the Rocky Mountain horse breed. Both of these genes are still being researched as they are not fully understood and are recent discoveries. Today's Ag Minute is about the gray gene in horses. The gray gene in horses is dominant, so if a horse has a gray gene, they will gray. Also, in order for a horse to be gray, at least one parent had to carry the gray gene. It's also possible for a horse to be homozygous for gray, which means all of the offspring of such horses will always go gray. Gray will occur on top of what other color genes the horse carries. Gray slowly removes the pigment from the base color, no matter what other genes are present. The horse will be born his base color, and then will begin showing the gray gene within a few months. Some horses are even born with a few gray hairs already present. There are many different shades of gray. Gray horses generally lighten with age. If the horse has a dark base color, the horse will often appear steel or iron gray early on. Chestnut with the gray gene will also cause a horse to be rose gray because the rose hue is still present. Dapple gray is usually the next step. As the horse continues graying, he'll go through the flea bit stage, which is white with little ticks of color. Lastly, a gray horse will lose all pigment and appear white. Today's Ag Minute is about the roan gene in horses. Roan is a color pattern caused by white hairs mixing with colored hairs. There are no roan-colored hairs on a roan horse. Roan is also caused by a dominant gene. Roans are often confused for grays. However, roans always have a dark head, legs, mane, and tail. They do change from season to season, but the head, legs, and tail always stay colored. Some roans may get darker with age, whereas grays get lighter. Also, when a roan horse is injured, the hair will generally come back as the darker base color, not white as in with other colors. When the roan gene appears on a chestnut horse, the horse will be a red or strawberry roan. The genetically correct term is chestnut roan. Likewise, the gene added to the bay base color results in a bay roan. Sometimes these horses are called red roans. The blue roan color occurs when the roan gene is present on a black horse. The correct term is a black roan. Roaning can occur with other gene combinations as well, so it's possible to have a buckskin roan or a dun roan. Previously, it was thought that the homozygous form of roan was lethal and that mares aborted the fetuses early in pregnancy. However, this is not the case. UC Davis has documented several instances of homozygous roan stallions that are alive and well and producing 100% roan offspring. This concludes most of the solid colors in horses. In a later week, I'll discuss the genetics of the Appaloosa and Pinto color patterns. This is Stephanie Falck. Thanks for listening.